Okay. Great. Hope everybody had a good break. I did. My version of break is to spend a lot more time on OpenWorm, as it turns out. Some of you were like, I actually want to take a vacation, and I was like, what do you mean you can't come to my meetings over the vacation break? So sorry about that. Apologies for those of you who actually want to take a real break, but it's okay. It's good. Actually, some good stuff got done, and I want to talk about that. I have the agenda here that I'm pasting for folks. And paste for Balash. Coming in. Okay, yeah. I just want to make a few comments. I don't want to drone on very long, but there is some stuff I think as it's a new year, it's worth thinking about where we are just a little bit. So some highlights from 2014. This is surely an incomplete list, and I want to create a more complete list, and I'll talk about creating a more complete list as we go. But 2014 was a great year. I think this community grew a lot in 2014, both just in terms of numerically. I was, over the break, catching up and adding some additional contributors to our webpage, and we've hit 60 contributors, 60 legitimate, like either checked in code or helped us with manuscripts or gave us data. We've officially hit 60 over the course of our entire tenure, and that's really, really exciting that we've gotten so many people to work in the space of computational biology. It shows no sign of stopping. More folks continue to come in and continue to do great work with us. So some of the highlights of things that I've seen, in 2014, we had C302, which we didn't have before, sort of a more functional way for folks to be able to run the connectome, to play with different computational models. We had a successful Kickstarter, of course, which I think increased everybody's morale and gave us a lot of nice press. A lot has gone on in Geppetto. Many releases have happened in the last year, and that team has continued to grow, spawning its own sub-team in this last year. We had Google Summer of Code for the first year, sponsored 2 students to work with the project, helping us push forward on PyOpenWorm and expanding on the Geppetto recording format. We had movement validation progress, and we had that team grow. We had hard drives of movement validation data donated. I almost thought it went without mentioning, but it doesn't. We had our paper published, which is the sort of first in squarely within the context of OpenWorm, as opposed to the previous one, which was sort of pre-OpenWorm. So that came out online as well, and that's now gives us something that's citable. And there's many, many more things, and I want to go into more detail on them. But just as at a very top level, I think this was a great year for us. I think it was a building year for us, a year of expanding our infrastructure, a year of also dealing with scaling the group beyond the initial passionate members that originally started it. But I think that the excitement continues to grow online. We're up to 1,800 Twitter followers, which, in the Justin Bieber scheme of things is not very many, but in the what, you're doing a project on simulating a worm and you have 1,800 Twitter followers, actually it's pretty good. We're about to cross 1,000 likes on Facebook, which again, is I think a strong statement for such a very specific project as the one that we have here. And I continue to hear about folks that learn about the project not from me, which is always great. So that was 2014. Now, what's possible in 2015, I think, is even more exciting, and I'm really looking forward to this year. I was getting increasingly excited over the break, seeing the energy of a new crop of volunteers coming into what we're sort of calling the muscle neuron team right now. So I think scaling up is an organizational challenge. I think it involves more coordinators. In this year, Matteo really took the lead with generating an open Geppetto meeting, which is sort of been a sub-team under OpenWorm, is also related to other projects. And then also Michael Curie sort of stepped up to lead. Michael Curie and Jim Hokanson jointly have stepped up to lead the movement validation sub-team. And lots of us exhibit leadership just in terms of taking charge of a project. I don't mean to suggest that this is any more significant than any contributions that folks have made, but I think that the challenge of basically limited people in a hangout is that as we grow the project, as the project continues to attract folks that want to help, coordination of this group of people now about 20, 25 that are kind of regularly filtering through the project requires us to think about this in terms of sub-teams. So I think in 2015 we've already got a template for that. I think we need to continue to work on that template and figure out how that relates to everything else in the project, and obviously there'll be challenges with that, but I think we've already got a template for that, and so I think we can build on it. Of course, another thing that's very exciting is, and we already got the starts of this in 2014, is really closing the loop amongst the major sub-projects of the of OpenWorm, closing the loop with Geppetto, cybernetic, movement validation, C302, getting all these things combined, working together. Of course, we have sort of multiple strategies for this. Some of it involves packaging everything in Geppetto, and that is going very well. Some of it involves taking some pieces of this and doing some experimental prototyping outside of the context of Geppetto, and those have also led, I think, to fruitful explorations. And as we continue, these different sub-teams are going to do these different things, but I want everybody to just think of a day not so far in the future where we really are seeing these things all working together, where we really can combine the like the statistical strength of the movies with the work of cybernetic, and with the C302 neuron model that's already sort of somewhat plugged into cybernetic, and just seeing all these things put together. This is a very real possibility for us in 2015, and something that's very exciting. And another, of course, came from the Kickstarter and all the great work that we did in coordinating that work is releasing the worm sim. And I think we need to continue to think about what that means for us, but of course that is going to be a place where lots of folks who have helped to support and back the project can really see much more visibly all of the things that we've been working on. And so that's an exciting bit for this year. I think there's also the possibility that we get another paper out at least from the work of cybernetic that's been gone in. So that's very exciting. And so I think 2015 is very cool. I think there's a lot of good stuff for us to look forward to. So couple of things then just on scientific teams with coordinators as we're scaling this up. So currently there's a Geppetto team, there's a movement validation team, which has separate meetings from this one, and in the last 2 weeks of December, we kicked off a muscle neuron modeling team, which is sort of really an aggregation of folks that were both working on C302, C. elegans NeurML, as well as people that were working on ion channels and the muscle model, and kind of putting those folks in correspondence with each other, along with other volunteers that had expressed interest in working at this level. So with that, I just wanted to talk a little bit about the future and what that means in terms of the structure that we have been operating on. In this year, I think we kind of got in, because we've been transitioning from, I think, a smaller team to a much larger team. I've at least lost track a little bit of this concept of releases that we once had, which was to sort of bundle up all work every 6 months into a retrospective and a prospective plan. We still have milestones up on GitHub that kind of reflect what we're doing. But now that we're talking about and establishing these teams that have separate meetings, it seems like the appropriate scaling process is that each of these teams is responsible for reporting on some longer-term chunk of work than every 2 weeks, as well as defining for itself where it's going in the next 6 months. So something for us to think about, not necessarily a great length here. We can take discussion on this, offline, but I think it would be good for us to start to approach the process of doing that in a bit of a more distributed way. Of course, it doesn't mean that if you're not working in a sub-team that you can't contribute to this as well. I don't mean to suggest that at all. I think there's always going to be folks doing individual things that are, not squarely in a sub-team. But I think basically we've outstripped my personal ability to check in with every single person in the project and ask them what they're doing and what their roadmap is. We did some of this when we met together in London, and that list is available for larger roadmap for the year. And I want to put these things in correlation with each other. But I think that it's sensible for us as we continue to grow to work on thinking about releases in that way and asking a little bit of those teams to help put that together. So I will be hitting up Michael and Jim and Matteo and then myself as the current coordinator, I guess, of the muscle team subgroup to do that, and then see where we can sort of sort other folks in. Because I think we can build other teams around other sub-projects. The question is just who actually has the bandwidth to be able to, coordinate the other people. Everybody's doing great scientific work, and some folks have been able to go above and beyond and do coordination work. And as I know for myself, it's really a whole other thing, emails and all that sort of stuff, and setting up meetings and doodle polls and that kind of stuff. But as we go forward, it is probably necessary. So we need to think about that. And then that also brings to the next topic, which is we have been invited to participate in Google Summer of Code 2015 by INCF. They liked what we did last year, and they're already asking for ideas page stuff like now. So I've got an email from INCF saying, hey, where are your ideas? So that also goes to everybody here, and as well, I think, to the sub-teams, which is it'd make a lot of sense if we get new folks coming in, if they're already in the context of one of these teams. And so there's obviously natural stuff because we had one in Geppetto before, and then I would think that I don't know if High OpenWorm is its own team enough yet. There's kind of sort of building one yet there maybe. I don't know if it should be considered part of Muscle Neuron or maybe separate. I don't know. But anyway, I will also be asking folks to submit ideas for that and crowdsource those ideas for what we could have somebody who would start in the summer working on. Obviously, there's a nice correspondence between thinking about the plan for the next 6 months and then thinking about what somebody at the end of those 6 months would come in and then extend. So those things are kind of related to each other. So we were the only sub-project under the umbrella of INCF last year to get 2 contributors. We're very grateful to INCF for giving that to us, but that was based on the strength of the people who came in and applied, and also I think because we were able to put enough interesting ideas in front of those people that they were willing to apply. So let's see if we can even go for 3 this year. Or at least we should still have 2. That obviously brings a lot of energy into the project. Okay, almost done. With my updates, actually I'm going to move Muscle Neuron team to the end so that I don't just keep running on. So I'll update that at the end so I can be quiet. Yeah. So another important development that has happened, and it's still in experimental mode, but it's looking like folks are liking it. So I'm going to encourage it while I can. Is for a long time, we've been trying to find a good solution for chat rooms within the project, and we've had experiments with experiments with IRC that have only worked for very limited periods of time until everybody kind of went away. And this year, we have discovered, thanks to a contributor, pointed us to this technology, something that seems like it could be very powerful for us as we scale up and as we have projects and sub-teams, and that's this thing called Gitter. I've linked it on the agenda, and it's very easy for folks to get to. It has nice connection to GitHub. I'll show you actually. So this is the OpenWorm/OpenWorm channel, which is kind of intended to be the most general. There's not a lot here, but there's actually quite a bit that's already been going on the PyOpenWorm channel, which is tied to the repository. So already good conversations going there in terms of trying to sort out how to make it to the next the next release of this, lots of folks can figure out what's going on. As well, the muscle model group also has a lot of folks that are having discussions in there, and you can just see by opening this up things like, you can paste in screenshots, and you can reference GitHub issues and then get these nice little pop-ups that show you that stuff. You can do app mentions to GitHub users and get more information about them. And it kind of keeps the whole thread of conversation going in a way that I think the mailing list hasn't. So I would suggest that if you haven't seen this yet, that you go check it out, that you go join channels for the repos that you're interested in. I think that this could be a very powerful way for us to keep in touch in between meetings. So, yeah.
Could I just suggest that I've seen a lot of comments there, discussion on some of the things. I've even, I think, seen a few at P. Gleason comments in there. Mm-hmm. When I first saw this, my impression was, okay, there's another thing now to actually keep an eye on, keep following, keep up to date with. I think it's fine from a point of view of, okay, if there's some, keeping an archive of chats and discussions which may or may not be relevant for any given moment in time. But I mean, anything which is an actual technical question, technical issue, it probably would still be good to refer those always to GitHub issues, because I think most people are now used to dealing with them and can list them and can go back and look which ones assign them and so on. So, I mean, for me especially, I mean, I don't know how much time I'll have to actually just check through these, but if something is in an issue, then it will be a permanent record there that I can go back and just check through this.
Yep, that's good to keep in mind. This shouldn't replace discussion in an issue, but sometimes it can be quick to just get a quick answer to stuff this way. So, yep, and obviously if you're not writing stuff in the channel, and no one will expect that you are there. It does. It will for stuff that mentioned, and for channels that you're following, it will also send an email daily. But I know we also don't need any more emails. So yeah, again, it's an experiment. No one's forced to use it, but some folks are really liking it, and I am going to take the chance to.
Yeah, no, it definitely looks good, but I think it's if there is something concrete which needs to be done, which needs to be addressed and finally closed, then yeah, definitely an issue would be the best place to actually discuss it.
Yep, for sure. Agreed. Okay, and then last thing before I hand it over. It was there was a suggestion for this meeting to do one thing. So we talked last time about making this monthly. So we have. So the next meeting is at the end of this month. So the format of this, though, the structure that we've typically had is that we kind of all go around and we do our quick updates about what's been going on, and I think we should keep that. But it's also been suggested, and I kind of like it, and I'd like to try it out, for us to actually have one of us actually go a bit more in depth on what's been happening in a longer time scale, more than just in the last month maybe, or maybe just more than just describing something verbally, but to actually do a bit more of a show and tell, and to kind of get a bit more in depth in the part of the project that they've been working on. So the suggestion would be that we kind of go around every time we meet, and there's a different person who will spend about 15 or 20 minutes of their time to do something. Could be like tutorial, could be a demo, of what's going on that allows folks to ask questions and really play with it, and something that's a bit more interactive on a single topic, and then we'll do the updates otherwise. I think it'll help to make this a bit more interactive, and I think it'll also help us for this idea of integration, for us to really get a bit more sense of what's happening in somebody else's world than we do now with just the quick updates. So would anybody be up to volunteering for doing that for next time? We would put it into effect next time.
I'm happy to do that if you want.
Okay, great. So we'll give you, I'll try to keep my comments shorter for the next time, and we will give you more like 15 or 20 minutes. And if you want to just like do a walkthrough of GitHub, that's great. If you want to do slides, that's great.
Maybe a show and tell kind of thing.
Show and tell. The emphasis would be like let's see code working, let's see diagrams, let's see pictures, like let's see like stuff like happening, so that also folks that are watching can like maybe get in to some repo if they haven't, with an eye towards folks that don't really know. That'd be great. Thank you, Michael. I will put you down. Okay, and I will be quiet. I will give an update on Muscle Neuron Team at the end. I will let the process go forward. Looks like Movement Validation Team is up first.
Okay. So Happy New Year, everyone. The Movement Validation Team has been pretty busy the last month or so. We had our first standing meeting on the 14th of December, so hopefully we'll continue to do that on a monthly basis. The next one is on the 14th of January, so feel free to join that if you're interested in listening to this. So in the last month, we've ahead of that meeting after the meeting that we had in December, we basically discussed that we would try to focus on getting a minimum viable product, so something that really closes the loop on the Geppetto team's work, so that we can actually provide that fitness function, however basic it is. So that's sort of been something that we agree on is what we want to focus on. So we have about 4 at this point individual team members that are active and contributors. So the last month, Jim's been working on computer vision heuristics, trying to extract the good work that Ev put into his computer vision algorithms, since we're choosing not to actually use their specific algorithm, at least taking the best parts out to try to use in ours. And he's also, you've probably seen in some of the updates, he's cleaned up a lot of the issues in GitHub to make it clearer for new contributors. Joe, a new contributor, Joe Bowen, he's been very, very active, so it's been fantastic working with him. He's made major progress on the cloud-hosted version of movement validation, and this is an important piece of being able to close that loop and provide that fitness function for Geppetto. So we have a new repository for that, and I linked to that in the agenda here. And I think he's also worked on several things outside of movement validation. So anyway, seems like he's doing a lot of stuff. So that's fantastic. For myself, I've put together a database schema that accommodates multi-worm videos, which is what we're expecting to get data for out of the Brown Lab. Now that's not quite in the minimum viable product, but nevertheless, just the basic database schema will be useful for the cloud-hosted version of movement validation, which we're hoping to move everything to. And Chris Barnes continues to work on the computer vision algorithms himself. So we also have several potential new contributors, and we're trying to continue to engage with them, and hopefully they can make a contribution soon. But for now, these are people that are working actively on movement validation. And yeah, so I'm looking forward to the meeting next week where we can get fully updated on people's progress.
That's great. Good stuff. All right, so then let us continue. I'm excited to see stuff next time, by the way, coming from that. Let's continue and go on to the Geppetto update.
Okay, so yeah, there was a bit of a break as well in the Geppetto meeting. Some things happened, actually, very good things. So I will try, just in the spirit of keeping people entertained, I will try to share my screen and show what has become release 0.2.0, which is already deployed on live.geppetto.org. So I will show one sample that was added of a network. It's not the C302 network, because unfortunately with that one there is one bug that we still have to fix. But the functionality that I'm showing will be available for any network, so pretty relevant also for C302. Again, all this work was the effort of all the contributors that are working on Geppetto. The release, which is 0.2.0, is not on GitHub yet. I was like in the background. I was working to put it there. Basically, I was testing a bundle that I have, etc., etc. So, is my screen sharing working?
Yep.
Yeah.
I can see.
Yes.
So, so very briefly, this is a network of multi-compartment models, and if we remember the original network of the C. elegans that we have, what in the website is linked as the connectome, that's also a network of multi-compartment models. That particular one is not fully in NeurML2, therefore we cannot see it here in this like new fancy way like this, but it's a matter of updating the model in that case rather than updating Geppetto. And but the functionality that I'm showing will work as well for that one and for any other neural network for what matter. So things that get added is the ability to have these connections lines that you see that will make explicit the connections between the neurons, because before we had it only through colors. So depending on what color each one of the cell was taking, you would know whether that was an input, an output, or both. But now you actually see the lines, and it is possible to basically get into the details. Okay, it is obvious to see that the yellow one that is selected is connected to the brown one here. And I can then click on the brown one here, which it's clear that it is connected to this one. I can click that one, and I can basically explore the network in such a way. Another thing that got added, and special thanks in this case to Adrian and Boris, and two of the contributors for Geppetto for building this. It's a brand new widget, which is a connectivity widget. It is it is really the first version, and there's a lot of improvement. There's already 2 cards on GitHub for things that we've all seen can be better, but it's nonetheless already a pretty good beginning in the sense that it basically makes, in a very graphical and direct way, it shows how the different cells are connected and through which kind of synapses. It is possible to change the order that is used to group the different cells by using these drop down here. And I don't know, I get excited every time that something moves, but this is really this is really cool. And if you are interested in terms of what kind of things we want to improve, well, there is documentation. There's a bit of documentation still that could be, yeah. You see, these are the issues that we currently have open. The connectivity lines that I was saying that are not currently working with C302. The connectivity, and in terms of the connectivity widget, there's a bit of things here that would make it better. So as always, I just wanted to remind folks that through that basically everything is in waffle, things that can be improved, and if any of you will ever want to like add a suggestion or something like that, it's a card there. We'll see it and we'll work on it. So this that I'm showing here is what's in live.geppetto.org, is version 0.2.0, and I just wanted to show the highlight of something that people hadn't seen before in this OpenWorm meeting. There is a lot of exciting work that is starting now getting to Geppetto in terms of having the ability for using other simulators on the back end, other than the ones that we're currently already supporting, having the ability from the front end to set the value of parameters that will give the user the ability to change the model. And we're also starting to work on a feature that will allow every user to have their own simulations, and that's in part of with the integration with open source brain that already happened over the past month. and that now is using Geppetto to basically show any model that is there. And just as a reminder for folks that haven't ever looked at it, Open Source Brain is basically, yes, if you type the proper address, Open Source Brain is a resource for sharing computational neuroscience model. It's where we have, for instance, C302, our Connectome hosted, the muscle model. So basically, we're working together for basically bringing and creating a new tool, which is making working with the neuroscience model in this case much more interesting and aligned with the time in which we're living. And I will probably stop here.
Awesome. Very cool. So C302 loads by itself, right? But it doesn't load with this. So what's the bug that's?
Well, if you load C302, you will see that these connections lines that you could clearly see here, they are kind of in the wrong place and they are very short. So C302 still works. It's the connectivity lines that needs fixing.
Got it. Okay. Yep. Just wanted to clarify that.
Yeah.
Cool. Yeah, I'm looking forward to having those connections in there as well. Let's see our worm with connections.
Yeah.
Awesome. Okay, let's go to Giovanni on WormSim.
Hello, can you hear me?
Yes.
Okay. Happy New Year, everybody. So, Worms, pretty much main stuff I'm working on is the WormSim sub-project, which is the worm in the browser, for short. And now the use cases, docs been released a few months ago. Still in some more of a flux, but very minor. I mean, most of the functionality has been nailed down in terms of use cases, but as it gets implemented, improvements may happen. Well, that's pretty much why we consider it to be in flux. Could improve once the stuff gets implemented and you can use it, and you say maybe this could be better, could be improved this or the other way. Sometimes things on paper are different than using them. But yeah, development is underway, and there's a waffle board that you can filter for WormSim tag, and you can see all the stuff that's currently going on and the stuff that's lined up. And in terms of ongoing tasks, there's a number of people currently doing work on this. In particular, over the holidays, Jesus was giving us a hand and taking up some tasks. So in particular, stuff that's ongoing is in terms of streaming the recordings to the client. So the strategy that we took for the WormSim was to, for the SPH particle-based simulation of the mechanical simulation of the worm, since that simulation would be too big to run in real time, then we're doing a recording. And Geppetto needs to be able to stream recordings from the server side to the client side, and work is happening right now to troubleshoot this and pretty much make it work. A lot of work has been done by Matteo already, and Matt was working on that, and Jesus is now troubleshooting this part of communication between the server and the client. We are successfully reading the recording files, but we're pretty much now making sure that all the data is going from whatever file on the file system of the server gets read, and it gets streamed to the client and added to the runtime model in Geppetto. So that's happening right now. In parallel, Jesus is also working on adding tutorial capabilities to the WormSim front end. So we want to be able to guide people through what the WormSim is. And we're going to do this in a similar way the current tutorials that are on Geppetto. So we're going to reuse the same JavaScript plugins that we developed. I'm calling them plugins, but I don't mean that they are actually jQuery plugins. They're just a snippet of functionality that we're going to move in over to the WormSim front end. And this will enable tutorials to happen on the WormSim side of things. Another thing that's happening currently as we speak, in the last couple of days, we were able to deploy the WormSim front end to a demo instance. Currently having problems with that right now, so I really was looking forward to show everybody a demo of that. And in fact, we showed a demo of that yesterday at the Geppetto meeting, but the server is currently down, and Jesus is working on troubleshooting problems there. Apparently, it was just a problem having to do with cleaning up folders of the previous deployment, so versioning issues with bundles, but expected to be fixed at some stage today. Other than that, the team, other people on the team are Martin and Sergey. Martin is helping us with the extraction of the SPH particle-based simulation into meshes and skeletons for these meshes so that we can animate the meshes and stream those animations. And this is a work that has been happening in the last few months, and there's going to be a bit of a slowdown on that particular work, even though most of the stuff is done. There's still a few problems to solve. But Martin is on his honeymoon currently, so there's a bit of a there's not gonna be a lot of progress in January on that particular item. But it's not a roadblock for any of the other work, so we can still keep doing what we're doing and start working with the dummy data instead of the actual data that's going to be at the end. Also, Sergey is helping us with the WormSim stuff by helping us create an SPH recording that can loop. So initially we just generated a recording from a straight worm that then bends and starts wriggling, but we realized that it would be nicer to just have it already bent to start with, so that we can loop, the recorder can loop indefinitely and give an experience that is smooth and it never kind of stops or goes back to being straight. And then also Sergey is helping us kind of cross issue with Martin. Where, so basically Martin is working with data that's coming from cybernetic and extracting these meshes from the particle-based simulations, and he found that for a particular recording they was working with, there seems to be an out-of-sync problem between the cuticle of the worm and the muscle cells in terms of the shape that they take. So we're currently talking to Sergey about that stuff, and Sergey is still on holidays, I believe, so that stuff's going to happen next week at some stage. But yeah, so that's pretty much it. I wanted to mention that Balash, that is all here today as well, but doesn't have a microphone to speak for himself, also has volunteered to help with the drafting text for the WormSim tutorials, and already provided a draft for the main WormSim tutorial, and also volunteered to provide drafts for the domain-specific tutorial. So when we go from the WormSim front end, there's a generic tutorial about the worm and about what people are looking at, and then they can go either open and explore the crawling worm, which is the SPH recording, and there's going to be a tutorial there, or they can go and explore the worm, the C. elegans brain, which is the C302, at least the scaffolding of that. We're going to build on top of that. So we're going to build a tutorial for the C302 simulation, and Balash is helping writing that stuff up. And yeah, so maybe if you have access to the agenda, Balash, drop links to that. I don't have them handy right now. But yeah, so that's pretty much what's going on. And in terms of what happens next is that we need pretty much close the loop between what Martin has been doing. So Martin has been working with the output from Cybernetic that Sergey is providing. So we need to take this processed recording and build a Geppetto recording file. But in order to do that, we need to expand the recording format to allow for visual transformation. By visual transformation, I mean this animations of meshes based on skeletal animations. At the moment, the Geppetto recording format doesn't have anything to store the data. So we next thing that's going to happen, we need to expand the schema of the recording format to allow us to store the information. And once that's possible, the streaming will be possible because that's already happening right now. Jesus is working on that. So pretty much we'll be in a position that we have the recording file. The recording file can be streamed to the client. And Martin, another thing that he's been working on is helping us extend the client side of this to play the animation that's been streamed. So in the next month, by the end of February, we are targeting to have the worm and the browser, at least a demo, rudimentary. I don't expect it to be finished because our release date is by June 2015, and so far we seem to be on track with that. But I'll be surprised if by the end of February we don't have a rudimentary wiggling work in the browser coming from SPH. It might not be from an SPH record. It might not be perfect because of all these problems that I was talking about, like how to sync things, but there's going to be something moving in the browser that looks like a worm, and then we're going to build on top of that for the following couple of months up to the release date. And that's pretty much all of it. And I'm sorry that I couldn't show the demo today, but or is it back up? Wait, it seems to be back up. I just got a message from Jesus.
Yeah, I was just testing it.
Yeah, I'm trying to log in now, and maybe I can show you something.
The login, it's working. Like.
Okay.
Late breaking news.
Woo! I remember my credentials.
Just in.
Okay, I'm just checking that I can log in. And now I'll share my screen. Oh, cool, it's there. Okay, I'll share my screen. Share my screen now. Good timing.
Bodes well for 2015.
Yeah. Okay, so that's familiar with this initial screen. There's a bunch of text here that actually I'm going to replace just with something like what we have in the use cases, but it could be anything. It just explains what this page is, lets people enter their email and submit if they want more information, or if you have been provided with login credentials, you can go and log in. And what was the test one login, Matteo?
It's test1 at gmail.com.
Okay. Password?
Password?
Okay. So everybody who's watching this, they're probably going to be logging on the worms.
We're gonna get North Korea to hack us in about
get it crashed.
about 10 minutes.
But yeah, okay. So this is the interior. This is the page the user will see if they have already created the worms. When the first time, let's say, to simulate the experience, the first time the user logs in, they're gonna see this page that is broken. So this is meant to be, all these controls are meant to be below. But yeah, it's just an expanded color picker. So yeah, this is under development, so that's why. It should look more like the previous page. You can pick a color and they will change the color of the cuticle, as you can see. They can name it, whatever you want, and then submit it. We'll go to this page. So the previous page should look more like this one, but with the expanded color picker. Something happened in the current development version and it was collapsed. So that's something that we need to fix. And what happens from this page is that the user can look at this model, and at the moment it's just this, but one of the things that we are working on as we speak as well, there is a control that will let the user show different layers of the worm, so muscle cells or motor or nervous system. And yeah, we've seen a demo of that yesterday from Jesus, but this version that's deployed at the moment doesn't have that control. So that's happening with this page. And then there's two buttons. One, as I was saying, so when I was talking about the tutorial, the tutorial will happen on this page. Balloons will open on this page. Maybe this worm will face the user as we're experimenting with this idea of the worm talking to the user and things like that. So we can do all of that. And we'll explain what these buttons are. So this is Explore the Chlorine Worm, Explore the Nervous System. So Palash has helped with a version of that, of this tutorial on this page. Plus, when the user clicks on these buttons, then Geppetto, the user is redirected to a Geppetto instance. In here with custom WormSim UI. I don't know if this is working at the moment. It's loading, so let's wait and see if that works. But the idea is that from this page, we go to the Geppetto. Yeah, this Geppetto coming up, and it seems like a slightly. I guess it's loading. So yeah, I don't know. Seems broken. This particular one seems broken. But the idea is that there's going to be a WormSim logo down here, so it's going to be a custom Geppetto front end, and the classic controls that are in Geppetto are not going to be on this one. They're going to be slightly different, including a home button to go back to the previous screen. The other one might actually work. Let's try the C302 one. Something's wrong. Well, you get the idea. Now, in this case, as you can see, it's linking to the C302 simulation in Geppetto up here from the bar, and it should bring up the C302. So, but this particular version seems to be a bit broken. So much for the demo. But yeah, yesterday it was working. But yeah, so that's pretty much as much as you can. This is under heavy development right now, so we're getting close to the point that we can close the loop, and hopefully in the next month I have a lot more to show compared to this current version. So that's pretty much everything.
Yeah, it's a good baseline.
Yeah,
Great.
We've got till June.
Yeah, till June. And there's a lot more than this. It's just this was a kind of a last-minute demo. Oh yeah, they fixed the deployment, and we gave it a shot. And hopefully next time there's gonna be a fully featured demo with the closed loop between Geppetto and the WormSim stuff, and all the other things that I was saying that are not here.
Well, it's new stuff we haven't seen before, so that's good.
Yeah.
Some new stuff.
Okay. Now that's all from me.
I wanted to ask one question, just in the interest of integration. There's been this idea that the skeleton extraction process is somehow going to feed into extracting features from SPH, and I just wanted to ask if anybody can say, or extracting features into movement validation from SPH. Can anybody say anything about either end of that and where we are, and if anything needs to be coordinated with that?
Okay, I can try and speak to that. In terms of what we are doing in the context of the WormSim is starting from the cybernetic simulation, extracting a skeleton. That work is already done, and animating the skeleton to basically reproduce the same movement that was simulated with a lot of number crunching by cybernetic. So what I think would be useful for the movement validation side would be to once we do have completed this animated skeleton, because at the moment there is the skeleton, at the moment there are basically in a way it's already completed, as in the rotation of the different bones of the skeleton is also there, but it's a matter of, okay, how do we use it? How do we consume it? And that bit, the packaging, basically, it's what still needs to be worked on. In the context of the worm sim, the way that this will work is by having a collada file which will represent the mesh, the skinned mesh, so it will also have a skeleton with all the joints, and a series of transformations that will happen over those bones. And for these transformations, they will be stored in the Geppetto recording format because they will be streamed by the server, and that will cause the worm to swim or wiggle depending on where it is. And but in terms of the movement validation, one of the questions that would be useful to see to basically work at the intersection is what would be the ideal format for the movement validation team to that they would like to see in order to consume these, to compute the features on the skeleton that have been extracted from Cybernetic, because that's basically the other part of the pipeline, right, Mike? At the moment, you're working on extracting these features from the video, and the other part of the pipeline is extracting these features from cybernetic simulation so that then the features can be compared. So the question would be, if, for instance, you have a collada file, which is an animated collada file, because that can happen, would that be a good input for the box that will then extract the features for the simulation side of it, or what would be the ideal thing? And consider that we're in 3D here while you are in 2D in the video, so probably there has to be something that happens there.
Yeah, that's a fair point. As far as 3D versus 2D, I mean, we can. It's not going to provide a very good fitness function if the worm is swimming in a three-dimensional material, given that all of our control videos are the worm crawling on an agar plate. But that said, I guess, yes, it's all of our movement validation assumes a 2D environment, so we would just maybe project the 3D object that you're providing to us onto 2D, onto a plane, and then at least we'd be able to get something out of that. I mean, again, our just talking to Jim, especially, like he's big on just having some kind of minimum viable product where we can have something to work with and go from there. So sure, yeah, by all means.
Yeah. So
that's a good thing to start with.
Yeah. The thing would be, once we have this collada file packaged with the animation, then basically it's like one option would be to like record a video from this in a sense, but it's like in that case, I suppose the question that we need to understand is you're doing some processing on videos of real C elegans. If you had a video of cybernetic, which by the way we do, could that video be used, could that be fed into the same algorithm, or basically you're expecting a specific pattern in terms of what is the background, what is the size of the worm, and things like that? But
yeah, no, in a sense it would be even simpler actually to get a video that really simulated as if it was like a microscope looking at the worm, rather than the fully specified skeleton all laid out. If it was actually something that we had to apply a computer vision algorithm to, and then obtain the skeleton, that would be interesting. And in fact, we've had many debates about that. And basically, the assumption has always been that we would get a skeleton from you guys, and that so you would enter the pipeline one step below the control videos, which would need the computer vision step to be skeletonized. But we can accept it at either stage, basically. It would just be one last processing step if you gave us the skeleton instead. Now, of course, we've spoken about this before, the idea of us giving you a specification for what you would need. And to be honest, one of the stumbling blocks has been that the pipeline that we've inherited from the Schaefer Lab code is not 100% clean, because there's never a point where everything is sort of monolithic. It goes from video right to features without a clean break where there's a point where it's just the contour of the worm, and so it's been hard to find exactly what the features algorithms need. Anyway, so we can, whatever you can give us, we can start with that and see what happens.
Okay. Yes. Let me know when is the next movement validation meeting. I know it's probably buried in some of my emails, but as I said in the email, I will try to join that just to see if I can help the integration there.
Sure, yeah, and there's been a couple of people for sure, like Jim in particular, and also another guy who are thinking about that exact step of the process, like the simplest specification for the worm, which would be presumably what you'd give us, if not that video that you're talking about. So sure, sounds good. I'll tell you.
Okay, cool. Thanks.
Yeah, and while you guys were talking, I was trying to see if we had made an issue anywhere in any of the repos, recording repo, movement validation repo, or the top level repo on this topic, because I have some fuzzy memory that we did that. And
it should be.
I found, all I found was this, although I'm not sure if it's the opposite of what we're looking for. Save worm features for use in Geppetto. We're working on saving. I think that's.
Yeah, that's kind of that's the other end of the loop, if you will.
Yeah, but since there was back and forth on this.
Looks like I'm assigned to this.
Oh. So we could just really use a crisp issue on this that makes it clear, and then maybe a volunteer who comes in can be assigned to be at the interface between these two things, just so that we make sure that we make progress on it. In addition to you guys talking. So let's try to define an issue on some repo in the next 3 weeks or so that really crisply captures this thing, and we can track progress. Okay.
Sounds good.
Great. Okay. Let's go on to Andre.
Uh-oh.
Hi, guys.
Hey, Andre.
Happy New Year, Andre.
Thanks a lot. Well, I'm sorry, I'll begin with quite sad news. Unfortunately, tomorrow we received the answer from the journal, our 4th attempt to publish first PCI-SPH paper, and again rejected. Possibly I will
Bad news.
paste here the answer. It's quite short from the editor.
It's outside scope. So the last 3 have been, it's the wrong journal. We don't publish stuff like this. They haven't even reviewed it, right?
Yeah.
But
So, well, I
what's the
don't
virus
like it. Something strange happens. Well, the text and pictures are quite good. Okay.
Okay. All right. Fair
I
enough.
think about the situation, and all the rest stuff is good. I will try to share my screen. Okay, maybe this one. Okay, this thing is quite familiar for all of us. And here are 3 neurons. There are 6 of them, 3 in the front of the worm and 3 in the anterior and posterior parts. So these 3 are connected with mechanosensation, touch stimulus in the front half of the worm. Very interesting, because all other circuitry connected with these 3 neurons is well, there is no general explanation which tells about how all these things work. How this is, for example, connected with mechanisms which deal with the muscular system, with this sinusoidal movement, and how this is connected with processing of touch stimulus. Because if any of these three neurons are touched, it causes the worm to almost instantly change the direction of its movement. So if it was moving forward, then it will almost instantly start moving backward. And how this happens is unknown. Well, one of the good news was that I found for free the worm book online. I pasted a link in one of the discussions on OpenWorm pages. I was very glad to find all these chapters, a lot of materials, hundreds of pages with good examples, explanations, and so on. So now I'm reading, selecting the most useful texts, trying to take good examples or parts of them to add to my experiments with that neurons which I have mentioned. Okay, what will happen if... Oh, okay, that works. So this is the window of neuron simulator, and there are 2.5 neurons. Two of them are already with dendrites, and one of them is still under construction. So quite a lot of surprises when I started to experiment with them. Here I tried to reproduce sizes of neurons, diameters and lengths and positions of dendrites quite close to reality. Also, I have collected some data about electrophysiological parameters like resistances, sorry, like currents. No, there is one thing like inverse to resistance. One divided by resistance. I don't know English term for this. Okay, no problem. There are some parameters, not quite a lot, but I have collected conductance. Thanks, Vlash. Thanks a lot. Yes, there are conductances through membrane, along the axoplasm, through dendrite, and so on. Some capacitance of unit of membrane, square, and so on. So I have collected quite a lot of them from various recent papers. I plan to share this information soon because, well, I think a lot of people from OpenWorm team will be interested somehow. Well, what is interesting in all this? All the processes are going quite fast. I expected to see something like traveling waves of excitation from one part of the dendrite to another, but it becomes almost immediately. And like, well, here, for example, you have two neurons, and they are dendrites, and one gap junction between them. It was not easy to make it. I had to compile some additional file containing some parameters describing synapse and some ionic mechanisms underlying its work. But once it's done, it can be used. So the signal, I will try to zoom this maybe. No, not this one. Sorry. So here is gap junction. So the signal, when it comes to this place, for example, if we simulate the touch of this dendrite here, here, or here, as it happens in the real worm, the signal almost immediately passes to this gap junction and then jumps to another dendrite. And here, for example, we see that color somehow represents the activity, electrical activity. Here, excitation is already active, and here is not yet. We can see, for example, if we continue, or if we compare the colors of soma of ALM neuron and this one is AVM, and this one is ALMA. Colors are different, so different voltage on them, because it's still not passed from this place to this place. Only a few milliseconds are enough for all this process, for the signal to spread from one neuron to another. Maybe chemical synapses have some other properties, but with electrical synapses, everything is very fast. So maybe for a year or a year and a half, I expected that there are some complex processes, some waves, like here is a signal, and then it decays, it travels somehow between synapses. But well, now it seems that we can consider any neuron with quite long 1 millimeter dendrite like some single entity. So if neuron is active, then everything is active. It's near its.
Yeah, I think that's one of the important things that I mean, because the neurons are actually so small that the electrical current, the change in membrane potential, will travel quite rapidly in the neuron itself. It's really just in very large cortical neurons like pyramidal cells where you will get slower movement of action potential from the tips of the dendrites through down to, or increase in membrane potential from the tips of dendrites down to the soma, and then potentially an action potential, and it will go through the axon very quickly at that stage. But in cells like this, it isn't strange that if you put an input into one location on the axon or the dendrite, for example, that it will fill up the cell very rapidly because it's just so small. You can obviously change the passive electrical properties of the cell, the axial resistance, and so on, the membrane capacitance, to change that. But I think the thing is, if you're trying to find the actual electrical properties from the literature, if you can find them, that's great. But it's probable that, yeah, there will be a synaptic input will rapidly spread through the rest of the cell. The one thing which may be more slowly traveling within a given neuron is maybe the calcium concentration, and that might be also an important factor, because a change in calcium concentration will travel a lot slower. It's still an open question about how much it might travel up any of these dendrites, but that can have an important impact on the behavior of the cell. So yeah, I mean, the ultimate goal is to incorporate all this detail into the models. But yeah, I mean, it's good that you're doing this manually. But yeah, I know it looks like a very impressive start.
Thanks. By the way, here I have only electrotonic gradual potentials, no action potentials, and so I have inserted all parameters which I have found, which describe C. elegans neurons. So everything is as much realistic as it can be extracted from recent papers.
Okay. How did you construct the morphologies? Was that by hand?
Here, yes.
Okay, because all of these are obviously in NeuroConstruct and NeurML already, and can be generated in Neuron from the NeurML, which was originally generated from the Blender files. So if you have NeuroConstruct running, you can potentially generate a simulation in Neuron. You should get a neuron morphology file for ADAL, ADMA, and various other neurons, and you can potentially use those. They may or may not fit this particular morphology. They will actually be following the curvature of the C. elegans as is shown in the 3D full neural network that we have on various images. But I think it might be a good starting point as well if you just want to connect up a small number of neurons and pass current through them and experiment in Neuron. They will produce correctly sized and correctly shaped neurons.
Thanks. Very good advice. Well, I was going to use, of course, already existing geometry. Just was going to try a few neurons for the beginning, and just to get familiar with basic concepts, how it is made inside neuron, how to do it manually, all this junctions, synapses.
Okay, well, it is very useful to actually, if you're going to use Neuron seriously, to actually learn to code at the command line so that you can see what the structure of the simulator code is like. A lot of the stuff we're trying to do automatically generates everything, and if you don't know what's in there, it's very tempting just to treat it as a black box. So it's good to actually get your hands dirty with Neuron and yeah, get a feel for it.
By the way, am I right that I can take morphology, but I still need to manually add synapses and electric junctions, or they already exist somehow in that model?
In the NeuroConstruct, they're a little bit out of date, but the plan with C302 is to extend this to using detailed morphologies also. So the idea would be that you have a Python script which says, I want to use AD or AMA, I want to use AMAL, I want to use the connections that are known to exist between those, and I want to put a current input onto section X on ADAL. And you write this in maybe 10 lines of Python, and then C302 will generate that code for you, and then that can be run in Neuron, so on. So hopefully, I mean, I'll try to aim at a scenario like this, targeting some of those neurons where an equivalent simulation to this can be generated through that framework, and hopefully get exactly this connectivity in Neuron that can be played with and compared to your simulation here. And obviously update the values for the passive electrical properties based on what you've found.
Okay, great. Thanks, Valery. Are you okay if I will contact you soon about some questions related to all this?
Okay, yeah.
I cannot spend much more time because I already took quite a lot. So that's all now from me. Thanks for listening.
Cool. That's cool stuff.
Cool.
Intuitions about neurons. That's good. All right. So then we got 10 minutes left. Let's go on to Porig.
Okay. Not too much of an update of anything new. I've been, as Stephen was mentioning, one of the people who's been trying to take as much time off during the Christmas, so I had a nice 2 weeks off. But now getting back into everything, and there's a ton of emails I still need to reply to, including a number of questions from the Neuron and Muscle team. So I'm hoping to get to those in the next few days. I have attempted to set aside most of next week for OpenWorm and C302 related things, or at least NeuroML software development, which would be related to this. So I'm hoping to make significant progress next week, but we'll see what else crops up in the meantime. Just to some of the issues that were mentioned there, yeah, gap junctions, muscles have been added to the latest version of C302. As I said, I wanted to try to extend this with not just using point neurons, which are integrate fire, but now that the muscle and neuron simulations are a little bit more well tested, try to use some of the ion channels which are being developed there into some of the muscle model, make single compartment conductance-based neuron models, maybe incorporate the values for the muscle model into C302 and put this into the whole framework. Also with the view of extending it up with a new set of parameters for multi-compartmental models. So I'll see how much of that gets done next week, but at the very least I'll update what's there, try to update the various versions of JNeuroML that are being used, and make sure it's as stable as possible, hopefully improve the documentation. But yeah, build on it next week and try to be a bit more communicative with people about what's there, and try to bring in the latest from the muscle modeling group.
Okay. Very good. It's good that you're fresh and relaxed. So, and thanks so much for being able to support all those crazy folks that are coming in. I think it's to the benefit of NeuroML as well as to OpenWorm that this stuff is getting used. So, very cool. Let's go to Balash, who I guess doesn't have any microphone, right? Yes. Giovanni already said mostly what I worked on. Okay, I see there's some typed in stuff here into the agenda. I also spent holiday learning Python. Cool. Wants to introduce effect size and confidence intervals, alternative p-values. Want to use an excuse to learn Python. That's good. And you also say in the agenda you're working on the tutorial text for WormSim, which is cool. Yep. Okay. And you added the link, so folks can go check it out, see how it's going. Looks like it's a Google Doc. That's great. Okay, and that's all from you. All right, so then I will wrap us up then real quick with updates from the newly formed muscle neuron team. There's about 7 to 9 folks that are regularly participating in this group. Its purpose is to aggregate detailed biophysical information from articles and data sets into the simulations of the muscle model and the nervous system connectome. I've linked in the agenda to 2 agendas that of this meeting that have happened. They have been so far less about coordination and more about education. So I have walked through for the first time in an on-air hangout an intro to Hodgkin-Huxley equations and excitable membranes for folks that are just coming in off the ground floor without knowing anything about computational neuroscience. That was the first meeting. And then in the second meeting, I got into more details about how to apply what we know about the math into the actual muscle model repo and how to work with NeuroML and LEMS to some extent, and also trying to very smoothly transition into the stuff that's already happened within the project. So far, I've been able to kind of flesh out what work Rainer has done with the muscle model, and next time I intend to go more into detail on C302. There are folks that are working on both aspects. So anyway, but there are YouTube videos for both of those if you want to catch up. One of the neat things that has happened since this began, again, Joe Bowen, who has apparently been very prolific in the last few weeks, not only in this project but also in movement validation, I understand, created a intro to NeuroML and LEMS via a Hodgkin-Huxley model, which is up online and I link to as well, which is quite nice. And it basically goes from the Python description of the Hodgkin-Huxley equations down here, so just one Python script that plots Hodgkin-Huxley dynamics, to this example that comes from NeuroML 2 for describing a Hodgkin-Huxley cell using NeuroML 2 and LEMS. And then this tutorial then walks through line by line, understanding the capacitance and the ion channel variables and how they all kind of fit together in this content. So this this might be helpful for many of us. Actually, the work that Andre was just showing, you might want to look at this as well, Andre, as a way to kind of get a sense of how the math fits into this XML language, because it's actually quite nice. So that's one tangible thing that's already come out of this, and of course the muscle model is essentially a specialization of the Hodgkin-Huxley equations, so that's then one step away for folks to be able to understand that. Next things that are happening, so this meeting isn't standing yet. We don't have a standing meeting for this, but we probably will get one pretty soon because it looks like it's a pretty stable group. There's been a lot of conversations over the chat room, like I was saying. So we will be having a journal club related to the paper that describes the biophysics of the first synapse onto the muscle cell from motor neurons, so that we can better understand how to go beyond a simple synapse model and have something that's more detailed. We'll be doing that in the next probably 2 weeks or so. So watch out for that. And there's actually some biologists who aren't as much on the coding who will be joining us for that. I will present that.
Oh, whose paper is it?
It is it's this paper that we've sent around a while, but let's see.
I think another one, yeah. It's Samuel.
It's the one where they did optogenetics on, let's see, graded synapse. I'm just pulling up right now. It's,
Is any of the authors joining on that?
No, we don't have any of the authors. This is a pure journal club. Although we could try to invite them. Here, I'll just share my screen on it. It's and then I'll link to it as well. It's called Graded Synaptic Transmission at the C. elegans Neuromuscular Junction. Basically, yeah, they do optogenetics stimulation of the motor neurons onto a muscle cell, and they record from the muscle cell, and they see how stimulating either acetylcholine motor neurons or GABA motor neurons, how they affect the muscle cell over time. So yeah, so different light intensities have different stimulus on the muscle, and so they can decouple the two populations from each other, which is actually quite nice. So they can see the difference between the acetylcholine synapses and the GABA synapses, and yeah, I'll walk through it in a lot more detail. But yeah, it's these guys. It's from 2009, and it's yeah. So the other paper I think you're referencing is just about the muscle cell. This one is actually about the synapse. So quite nice. Okay, but yeah, good idea. I'll see if I can get the authors actually. That would be good. Okay, and then the other thing I want to say about the team is that because they are using NeuralML2 and LEMS, we have this nice ability in terms of integration to form a Geppetto user group, which I only kind of realized yesterday after sitting into the Geppetto meeting. But you know, there's a Hodgkin-Huxley example is the main Geppetto example, and it's run off of a NeuralML2 LEMS file. And the muscle model has also been almost put in there as a NeuralML2 LEMS file. There's a fork of the samples that Dan Kurchunin created of that. And in addition, you know, Geppetto lets you, the live version lets you just point it at some Geppetto file that has in some GitHub repo. So in theory, this could be as a user group for Geppetto, which I think has a lot of benefits. Number one, early feedback for features that we could incorporate into WormSim that other folks could use. Early feedback on how good the documentation is for some of that stuff. Ability for folks online to, you know, revise NeuralML2 LEMS files and then, you know, load up Geppetto and see, you know, how they work. You know, it's probably easier in some cases for them to use, you know, JNeuralML and that whole stack and all that. But you know, if for sharing that kind of stuff with other folks very rapidly, this provides at least a nice way for demos to be created pretty quickly. And you know, anytime we've got a way that these two groups that are coming from different places can talk to each other, I think it's a really awesome opportunity for growth. So I'm very excited about that. I'm excited about continuing to encourage this kind of crosstalk, as well as that group is also working with spreadsheets of ion channels and looking for ways to go back and do optimization to tune up models of ion channels, and that syncs in with some of the work of open source brain and syncing up with Channelpedia type work. So the group has been looking over at Channelpedia and trying to see how we can, you know, integrate stuff from there. So that's an intersection point. And then yeah, improving the, you know, the muscle model itself and making it actually have the matching what the Neta-Cohen model you know, says, and which of course then we need to continue to improve. But that's kind of what we're doing. So currently the project is solidify the muscle model itself so that it matches the paper, start to build out the synapse, and then start to describe the ion channels in the six or eight motor neurons that project to that muscle cell, moving beyond Hodgkin-Huxley dynamics into you know, analog dynamics, you know, using ion channels that we can infer are in those motor neurons. We don't actually have a precise list, but we have some ways to do some pretty reasonable inferences to figure out at least some of the ion channels that should be there. And you know, we set that scene up for that much in NeuroConstruct probably a year and a half ago, and you know, we had challenges building a team around lots of people, you know, working with that, and then so I think now with the NeuralML2 technologies, there's new life that's been breathed into you know, that effort. And we can do, and you know, there's other work with other subnetworks as well. There's still the social network that's a higher level interneuron network. There can be work done with other pieces as well. So yeah, so Mateo's got to go, and we all do as well. I've run over time. I had a brief meeting with the DevoWorm people as well. They're still in the mix, FYI. I can update on that more fully next time when I have time. But let me break here since we're out of time. Thank you everybody for the updates. Excited for 2015. This will happen again at the end of January. Watch the calendars for that. It's not the regular 2 weeks cycle. So
thanks.
You've open work.
Everyone.
See ya.
Bye.
Bye-bye.